Good afternoon. Welcome back to virology. So last time we talked about how viruses evolve. Today we're going to talk about how new viruses emerge, emerging viruses. So the definition of an emerging virus is the causative agent of a new or previously unrecognized infection. So it could be a virus we haven't seen before, or a virus we know but now causes a different disease. This term became popular in the 1990s. I believe it was coined by Steve Morse right here at Columbia. But emerging viruses, of course, are not new. They have always emerged. We have just gotten better at detecting them. However, I said viruses have always emerged. In humans, they really have emerged more frequently since the rise of agriculture. About 11,000 years ago, human populations got big enough to sustain the introduction of new infectious agents. Because before agriculture, human populations couldn't get very big because they didn't know how to feed them. But once humans figured out agriculture, which includes not just crops, but raising animals, then they could congregate in large numbers and a virus could be introduced and it would spread in the population. The press is fascinated with emerging viruses because they like drama and tragedy. So some other definitions for emerging viruses. As I mentioned before, it could be an existing virus that does something new. And another way to put that is the virus has an expanded host range with an increase in disease that we didn't see before. So that disease did exist. Now, a word that you need to remember is zoonosis. Zoo means animal, right? A zoo. And a zoonosis is the transmission of a virus from a wild or domesticated animal to humans. Could be wild animals or could be your pet. Zoonosis. Sometimes these zoonoses establish a new human virus in the population. So for example, SIV moving from chimps to humans became a human virus. SARS-CoV-2 moving from bats to humans became a human virus. But sometimes cross-species infection can't be sustained. So Ebolavirus, we believe, goes from bats to humans, but eventually does not transmit well among humans, and the chains of transmission stop. It never becomes a human virus that continuously circulates among humans. Same thing, MERS coronavirus, from camels to humans. It doesn't transmit well among humans. It doesn't acquire the mutations that allow it to become a human virus. Same thing with West Nile virus. Every human infection is a zoonotic event from mosquitoes to humans. It doesn't spread from human to human. Another name for zoonotic infections that's often used is spillover, right? The idea that viruses are existing in animal reservoirs and somehow they spill over into humans. So I may use that term as well. So that's Fever is the book that made me want to become a virologist. So you should read it and see if you've got it in you. This is the story of the spillover of Lassa virus from mice, from rodents to humans in Nigeria. It was the first zoonosis, I think, that was discovered. It's very nicely described in this book. All right, now we know the origins of human pathogens that are circulating today. And that's shown in this chart. So on the y-axis, we have viruses belonging to a number of genera. So the number 31 genera. Viruses belonging to 31 genera are human viruses. They once were animals. So all viruses that we have today came from something else, either a human or ancestors to us. 37 genera of viruses are zoonotic pathogens. They spill over and don't cause sustained infections. The next one, members of 16 genera, Homo sapiens acquired from ancestors of Homo sapiens. And then viruses from 6 genera we acquired from other Homo species, like Homo denisovans or Homo neanderthalensis, because we coexisted for some time with these other Homo species and we got their viruses, all right? So that is what we know about the origins of human pathogens. Now, what drives the introduction or the spillover of viruses from animals into humans? And I think the main driver is shown here on this graph: world population growth. So that is the numbers of people in billions over years, right? And so you can see that the human population initially was quite low. We lived in bands of hunter-gatherers of a few hundred people at the most. And then it grew and grew with the rise of agriculture until look at the explosion by the 2000. So what does this matter? Well, we change ecosystems, we invade ecosystems, we invade every part of the planet. And so there are already viruses in these parts of the planet, so we get them. Every spillover is our fault, okay? It's our fault. It's not that we manipulated anything. Well, sometimes we do manipulate the environment, and I'll tell you how we do that. But when a new virus emerges, it's something that humans have done. It's due to anthropogenic activities. And I'm going to give you some examples of that in a bit. It's our fault, folks. If we remained as hunter-gatherers, we'd be okay. But we invade everywhere, we trash the environment, and we get viruses as a consequence. So here are some of the activities, the ecological and anthropogenic activities that promote emergence. I love to say humans continuously devise new ways to encounter viruses. Of course, transportation is a big one. This is from that website where you can see every plane that's in the air at any given time. This is very cool. You can click on the plane and know the flight number, where it's going to, where it's coming from. You can see there's a lot of activity, and that brings viruses all over the place. But other things like building dams, irrigation systems, deforestation, wildlife parks, even things like blood transfusion and transplantation of organs from different species, like a pig. Urbanization, daycare centers is a big one. When I was a kid, there were no daycare centers. You had to deal with your kids on your own. But daycare centers now bring 50 kids together to share and exchange their viruses and bring them home to dad and mom, right? That has contributed to virus emergence as well. Millions of used tires. Remember, these are put on ships and shipped around the world. They're full of water. They have mosquitoes breeding in them. We have a mosquito in the US, Aedes albopictus. It was not here before it was brought here on a container ship with tires on it. It went into Houston. We know exactly when, and now it's spread throughout the US. And of course, climate change, even natural disasters, hurricanes can change wildlife patterns and change virus propagation. So humans do a lot of things to encounter new viruses. And there are plenty of viruses out there. Here is just a map of the Amazon North region of Brazil. So here's Brazil over here, and the North region is to the north. This is all being deforested at an incredible rate. So there are people working in there, cutting trees down to make farmland. All these little words that you can't read are viruses that we know about that exist within the Amazon North region. 183 arthropod-borne viruses, and other vertebrate viruses as well. So we are susceptible to infection. We are potentially susceptible to infection with those viruses. Any of the people working in there can get them. And that's not just the Amazon North, it's the whole world. So here is a table of some viruses that have undergone in the past zoonotic events. Some of them are still zoonotic event, others are no longer. And it tells you the virus and the factors leading to emergence. And so today we'll talk about some of these, but HIV-1, which we will talk about next time, entered humans through the hunting and butchering of primates for food. So in many parts of the world, we can't walk into a supermarket and get meat. So you go hunt it, and primates are a source of meat, and they have viruses, and that's what happened with HIV-1. Coronaviruses, the MERS coronavirus, which we'll talk about. This comes from camels to humans. So the practice of camel husbandry. In many countries, camels are pets, they are food, they are race animals. So a lot of people have camels, and so we get their viruses. And SARS-1 spilled over from bats into humans in open air meat markets or live animal markets, as did SARS-CoV-2. Notice it emerged in a live animal market. It did not emerge in a lab. No matter what anyone tells you, SARS-CoV-2 did not emerge in a lab. There is zero evidence for that. It's just rumor and innuendo, but we have good science data that the virus came from a live animal market, which we'll talk about in more detail next time. All right, so evolution plays a big role in viruses spilling over from animals into humans, right? In the animal, the viruses exist as a quasi-species. So every species of animal is infected with many different kinds of viruses, just like we are. And they exist as a quasi-species, right? This incredibly diverse collection of variants, and within it may be a variant that can infect people just by chance, right? Just by chance. It's totally random. So the presence of a quasi-species in animals allows them to infect potentially new hosts, right? That's variation in selection, like we talked about last time. A bat harbors a quasi-species of coronaviruses if it should encounter a person, and that's a rare event, right? It may give that person its collection of viruses, among which one may be able to infect it, but it's a very rare event. That's why we don't see new viruses emerging every day, because it requires a number of circumstances. All right, so before we talk more about emerging some examples, I want to talk to you about the different patterns of host-virus interactions that we know about, because this will inform us about zoonotic viruses. So here we have a series of boxes showing you 4 different kinds of host-virus interactions. We have one that we call stable. That's like influenza virus infections of humans. It happens all the time. It's a stable system. And many other viruses do that as well. Then we have an evolving system where the virus from a stable relationship goes into another species, and when it's initially there, it evolves very quickly as it adapts to the new species. This could be the same host, it could be human to human, or it could be animal, non-human animal to human. A dead end, one-way passage to a different species, like West Nile virus from mosquitoes to people. It's a dead end. Many other examples of that too. And the resistant host, so in the dead end, the virus will reproduce in the dead end host, but will not spread to another. In the resistant host, infection is blocked. The virus gets into the host, but there is no infection. Now, these are related to each other. The stable can contribute viruses to resistant, to evolving, or to dead-end hosts. So the major pathways are these two big red arrows. So the stable systems can contribute to dead-end or evolving systems. Then there's a little bit of interplay among the others as well. Okay, so let's look a little bit more deeply at each of these. The stable host-virus relationship. Both the virus and the host survive for the most part. They're not extincted. The virus doesn't go away. The host doesn't go away. And some of them are permanent as far as we can tell from our little view of history, like measles virus in humans. It seems to be with us. Herpes simplex viruses, the other herpes viruses like cytomegalovirus and others that we talked about. And now SARS-CoV-2 is a brand new virus that seems to be effectively permanent. It is not, humans are not the only host for SARS-CoV-2. And that's the last bullet point. The stable may include infection of more than one species, not just humans. SARS-CoV-2 infects many different wild animals besides humans now, providing a reservoir for reintroduction into humans or introduction into different species. Influenza viruses exist in many, many animals, including aquatic birds. Flaviviruses, togaviruses all exist in many other species, but they are also human viruses. So being human doesn't mean you're just in humans. The evolving host relationship is characterized by instability and unpredictability. And so in other words, you have a virus that's in a stable host relationship, spills over into a different species or the same species, and you have, that's never seen it before, and you have this evolving situation. So examples include introduction of smallpox and measles virus by old world colonists who came from Europe. They brought those viruses to the Americas, and they introduced them to the people living here. And here, they had never seen those viruses before. They had zero immunity. Even though they were humans, the introduction of those viruses into that population caused extensive spread and death. West Nile introduction into the Western Hemisphere in 1999. Same situation. We had never seen West Nile virus ever in the Western Hemisphere. So when it got in, it found a population of non-immune people and spread rapidly across the Americas. And the same thing with myxoma virus, right? You introduced into Australia to eliminate rabbits. Those rabbits had never seen myxoma virus before. So it spread rapidly, and in that first year kills 99% of the rabbits. Okay, then a dead-end interaction often happens when you have a cross-species infection. So it goes from a stable to a host that cannot transmit it to other hosts of the same species. No sustained transmission. And examples are Ebolavirus infections of humans, chimps, and gorillas, which are not the natural hosts of this virus, and the virus does not become adapted to them. Influenza H5N1 virus in humans has not become a human virus. It does not transmit, if at all, among humans. And now it's infecting many, many different kinds of other animals, mammals, birds, and so forth. So we are worried that it may acquire randomly the ability to transmit among humans, but it hasn't done so yet. And people who are alarmist about it are being alarmist for no reason, because there's nothing we can do about it. We cannot eliminate H5N1 virus from the planet. All we can do is do surveillance and see what's emerging and be prepared with antivirals and vaccines, which we have. I saw a guy on NPR the other day. This is a huge, serious problem for humanity. Why do you say that? There's nothing you can do about it. Why do you say it and scare everybody? It doesn't do any good. He wants the publicity. That's the end. In the end, when scientists say crap like that, it's because they want publicity. So these dead-end infections don't contribute anything to natural infection. They don't establish a reservoir. It's a one-off thing, and then that's the end of it. So here's some examples of stable and dead-end host virus relationships. So here at the top is the stable relationship. Here we have a virus that is transmitted among wild birds by mosquitoes. So this mosquito has bitten a wild bird, picked up virus, and then it will bite another wild bird, and this cycle goes on and on. So that maintains the virus in the environment. And chickens can often be bitten and replicate the virus. And sometimes other mosquito species will bite an infected bird and transmit virus to a different host that it prefers. All right, so that's the stable host-virus relationship. This exists for many, many years, and both the host and the virus exist. Now, on the bottom, we have dead-end hosts. Sometimes mosquitoes will bite horses or people. She's all covered up, so she's not going to get bitten. Well, she might, face, hands, you know. But if that mosquito has virus, it will deliver it to you. It may replicate in you. You might get sick. But it's a dead end. It's not going anywhere else. Why? Because even if a mosquito bit you, you don't have enough virus in your blood to transmit to another person. Now, horses can get very sick from these infections, and so people who own expensive racehorses go to great extents to avoid them getting infected, including vaccinating the horses. Here's another one. So this one would be a tick-borne encephalitis virus, which is transmitted among rodents by ticks. Ticks bite the rodent, they take a blood meal, and then they pick up virus. Of course, they go on to another rodent, and they're picking up another blood meal, and they're depositing the virus in the meantime. So this is maintained in a cycle among rodents for many, many years. Both the host and the virus survive, and the tick, of course, is the vector. Sometimes it can spread from tick to tick. The virus may infect the ova of the ticks, and then when the ova develop into new ticks, the new ticks have the infection. Sometimes ticks will bite a cow, which then has the virus in its milk. So that's not a dead end because humans drink milk, and so they can acquire the infection from a cow. Or the tick can bite people, right? Ticks, if you hike, you better be careful and look for ticks on your body because they can transmit virus infections, but they will not go to another human, again, because you don't have enough virus in you to spread to another human, even if you get bitten by another tick within so many days. So the humans are dead-end hosts for these infections. The stable hosts are shown above. Now, how do emerging infections occur? There are two steps. Encounter, right? We have some animal, a bat, with lots of quasi-species of different viruses. Nothing will happen unless the bat encounters another animal, and we're talking about humans here, so the bat would have to encounter a human. Actually, the bat could encounter another animal, which then would encounter humans, as we'll see happened before. So we have camels, rodents, bats, harboring quasi-species. By chance, they can make their way into people. If people are hunting bats, if they're doing things with camels, if rodents are living in their homes, they can transmit viruses to them. But going into one person is not enough, right? If it infects, somehow an animal gives a virus to a lone farmer who lives alone on a farm and never goes anywhere, the virus won't go anywhere. But if a farmer has a family and gets infected, he or she will transmit to the family. They may go into town and transmit to other people. So you need to disseminate. You can see it's a random process. It's a low probability random process. Again, that's why we don't see it very often. And so it happens a lot, we think. We think there are lots of spillovers that we never detect because they're in one person or a few people. They don't get very sick. They don't seek health care. That's the end of it. And we think there are many single host infections where individual gets a virus from an animal. Maybe it's an asymptomatic infection. Maybe they get mildly ill, don't seek health care, and that is the end of it. So we think these happen a lot. Maybe even here in New York City. We have animals here. We have squirrels and raccoons and other things, besides humans, of course. And so we can get viruses from them. We certainly can get rabies viruses from rabid raccoons here. And that's an example of a spillover. Now, when a virus comes from an animal into another host, like a human, it needs to encounter susceptible and permissive cells, right? So if it goes somewhere where there are no such cells, it will never go anywhere. Population density makes a big difference. The more people around that initial infected person, the better. The SARS-CoV-2 emerged in Wuhan, a city of 12 million people, a huge transportation hub. Perfect setting to spark an epidemic with lots of people. So would New York. And in fact, we have live chicken markets here in New York City where you can go in and pick a chicken and they will kill it for you, you bring it home and cook it. That seems to be a risk factor for H5N1. And the health of the population is also important. So eventually, for a new infection to establish in humans, you need a series of infections. You can't just have one or a few. You have to have a series. And that exactly is what happened with HIV and SARS-CoV-2, SARS-1, and many other viruses. But it's rare, right? Before SARS-CoV-2, the COVID pandemic, when was the last pandemic, viral pandemic? Do you know? 1918, but even closer than that. 2009, yeah, 2009 swine influenza, which I showed you how it emerged. And before that, 1981-ish, the AIDS pandemic. So they're not very frequent, and that's why you didn't remember them, because until 2009 is a long time ago, right? So they're pretty rare. Okay, so let's look at some examples. This is the fun part. Where did smallpox virus come from? You know, it's, at some point, millions of people died of smallpox on planet Earth. It was a horrible infection. It disfigured you. And so we seem to know where it came from. What we do is we look at the genome sequence of smallpox viruses and related viruses and look at their relatedness to each other. And it looks like the virus emerged 3,000 to 4,000 years ago. It crossed over from camels in East Africa here. So camels are not native to Africa. They were introduced to Africa 3,500 to 4,500 years ago. Camels originated in North America. And they went south and they went across the land bridge into Asia and Africa. And so they were brought to East Africa. And the idea is that they encountered gerbils that had the ancestor to smallpox virus. So that little cute gerbil had some relative of smallpox virus, gave it to the camels, it further diversified. And then because people are using camels a lot, got into people. And then it spreads. It becomes a human virus. It spreads all throughout Europe. And when the colonizers go to North America, they bring it with them, because nobody in North America had ever seen smallpox. How about measles virus? Did you know measles virus originated in a cow, a related virus called rinderpest virus? So rinderpest virus is a bovine pathogen, seems to be the ancestor of measles. So cows have always been infected with rinderpest virus. But you know, cows weren't always on farms. Okay, they were wandering around wild, and someone at some point got the idea to milk them and eventually eat them, so they brought them onto farms and domesticated them. And when population grew, boom, rinderpest went into people and became measles virus. it didn't explode until the population got big, because measles requires populations between 250,000 and 500,000 people to be able to spread properly. So once human populations got that big, the virus went from cows to people. It may have gone into people before the population got big, but it never went anywhere. But finally, when the population got large enough, it took off, spread everywhere, reached America by colonization and migration. Same for smallpox. This is an example of when a virus that's in a stable host-virus relationship gets introduced into a member of the same species that's not immune. So these are diseases of exploration and colonization. When a virus is introduced into a immune naive population, you have epidemic spread. So as I've already told you, smallpox reached Europe from the Far East, where we got it from camels, and then it made its way to the New World, where it changed the balance of human populations. It killed 3.5 million Aztecs in 2 years, which allowed Cortez, with his miserable band of 35 soldiers, to conquer the Aztecs. There's no way they could have conquered it without smallpox helping them. Some people think that he even introduced it, but I'm not so sure about that. These viruses not only change human populations, but they changed the balance of human populations and allowed Europeans to take hold in the New World. Another factor for movement of viruses from animals into humans is changes in human populations and environments. And I love this example of poliovirus. Poliovirus has been around for ages, thousands of years. There's an Egyptian carving from about 1500 BCE, which shows this priest with a leg that looks like he has polio. That's this very characteristic drop foot of polio, because your leg muscle is paralyzed. You know, normally when you lift your leg up, you can hold your foot up, but when they're paralyzed, the foot drops. That's a drop foot. And so there was polio throughout human history, but only sporadic cases here and there. No epidemics. Suddenly, in early 1900s, we start to see outbreaks of polio. First in Europe and then in the US, getting bigger and bigger and bigger. These are annual cases on the y-axis, thousands and thousands of cases. Why did it suddenly become an epidemic virus? So we would call this an emerging disease, because we never saw the epidemic form of polio before. Why did it change? It turns out that it's all about sanitation. Before 1900, polio circulated freely. We didn't have any toilets or sewers, so people would have pots in their homes that they would use to defecate and pee in, and then they'd throw them outside in the backyard or in the front yard. You know, the streets in New York were covered with human and horse excrement at one time. So this excrement is full of poliovirus, so kids get infected very early in age, shortly after birth, but they have maternal antibodies to protect them from paralysis. And so they get infected, they don't get paralysis, and they're fine. Now, in 1900, what happens? The greatest invention of humanity, sewers in the toilet. Now, the sewage all goes in pipes, and kids don't get infected shortly after birth. They get older and older and older. The maternal antibodies wane. Now they are not immune to poliovirus. And you introduce a virus into a few thousand kids, boom, you have an epidemic. So it is an epidemic, a disease of modern sanitation. Sanitation is not bad, but in this case, it delayed infection to a point where you didn't have immunity. Today, when you have a virus showing a new disease property, immediately scientists say, aha, it must be the genome, something mutated to cause this. Because that's the easy thing, the easy way out. You do the genome sequence. So in 1908, if we had genome sequencing, that's when poliovirus was first isolated, they would have compared the sequence pre and post-epidemic, found some mutations and said, aha, here are the mutations that cause epidemic polio. And they'd be wrong. It's not about that. It's about population immunity. But sequencing is very easy. When I was a grad student, sequencing had just developed. And the professors always, on tests, the professors always asked us questions. What is the solution to this without doing any sequencing? Because they knew we would just say sequence it and find the answer. They wanted us to think a little bit about it. I never forgot that. Here's another example of how changing climate makes a difference and causes spillover. Hantavirus pulmonary syndrome is a very serious lung infection, first found in the Four Corners area of Mexico in 1993. So that's where these 4 states join. You can actually go and see there's a plaque on the ground. It says Four Corners. And there were a series of young, healthy people who died of respiratory infections all of a sudden. They found the causative virus. It was called C. nombre virus eventually, and it turns out this virus is endemic in a mouse called the deer mouse, Paramyscus maniculatus. About 30%, and we have them here, 30% of mice are positive for the virus. By the way, when they first identified the virus, the CDC isolated it in a town called Muerto Canyon. So they were going to call it Muerto Canyon virus. And the residents of Muerto Canyon did not like that idea. So the CDC tried a few other names. They didn't like any of them. So they called it No Name, which I think is a nice show of how the CDC used to have a sense of humor. Now they don't because everyone's getting fired, right? Hantavirus is a bunyavirus, is an envelope virus with 3 ambisense RNA segments. So what happened here? Why all of a sudden did we get these diseases that we never saw before? These viruses had always been around. Hantaviruses were not new. So it turns out that in 1992-93, there was a lot of rainfall, more than usual. The increased rainfall made a lot of piñon nuts, pine nuts, which they're delicious. You can cook with them. They're very expensive. Humans like them, but also deer mice like them. So the mouse population rose because there were a lot of deer nuts. And as the mice population rises, the mice go in people's homes. And they're excreting virus in mouse feces. And you see mouse poop in your house. What do? You sweep it up. You aerosolize the virus, you inhale it, and you get Hantavirus pulmonary syndrome. And that's why it became visible in that year, because we had an abundant rainfall that increased the mouse population. Now, humans are not the natural host. This is not a human virus. It doesn't go from human to human. It goes from mouse to human. So it's a dead end. And we've known about this virus since 1959. We can see cases back then. Well, first, let me show you the distribution in the US. So there have been 831 cases of Hantavirus pulmonary syndrome from the 90s through 2022. And those are the states where you mostly see this. So the Western states, you mostly see them. And you see a few here. Here's the distribution of the deer mouse. It's pretty much throughout the US, except for the southeastern US, but we have it here in the US, in the Northeast. But also the other mice can carry the virus besides the deer mouse, white-footed mouse, the rice rat, and the cotton rat. So you should be careful if you have mouse feces in your house. You should spray it with bleach and let it inactivate before you wipe it up, okay? Now, this is what happened to Gene Hackman. So he and his wife lived in Santa Fe, which is a place where there is Hantavirus pulmonary syndrome. He had Alzheimer's. He was completely dependent on his wife for health care. She got Hantavirus pulmonary syndrome and died within a couple of days, never sought health care. And so he eventually died, I think, a week later, and they found them both dead, but it all started with an HPS infection. All right, so this is what happened with that. It was a very unfortunate situation. She should have probably gone for medical help, but she probably figured it wasn't serious, and then it got very serious quickly, and it killed her. And she was only 65. Okay, be careful with that mouse poop, all right? You can get it in New York State. There have been cases in New York State. Especially if you go camping on those wood platforms, you pitch your tent on the wood platform, the mice are underneath, and they will contaminate your tent. It happens in New York, it's happened in California at Yosemite National Park. All right. Now, let's talk about bats as sources of zoonotic infection. We did not know that bats carried so many viruses until this study done in 2011, which found that bats host major mammalian paramyxoviruses. Do you know what a paramyxovirus is? Measles virus, and also other zoonotic viruses called Nipah and Hendra. These cause severe disease in both domestic animals. They can also infect humans. This is a flying fox. He looks like a fox, right? Adorable. His wingspan is like this. Not so adorable. And they have claws throughout their wing, and if you catch one, they will try and poke your eye out with their claws. So they're not so nice. Well, they're just trying to defend them. So why would you catch one? Because many people want to know what kind of viruses they have. So they go to places where they exist, particularly Bangladesh and India, where there are lots of them, and they capture them. And one of them told me, he said, you have to be very careful because as you're sampling them, they're going to come around with that hook and try and hook your eye. All right, so the first bat-borne virus discovered was Hendra virus. This was found in a town called Hendra, Australia, in September of 1994, okay? Those red dots. It was an outbreak in a horse farm. It killed 14 racehorses and a trainer. So the trainers are working closely with the horses, right? Brushing them and all that. And so the horses got sick and the one trainer got sick. So what happened here, the stables are open air stables. At night, the flying foxes come in, they contaminate it with their urine that has virus in it, and then the horses get infected by that. This still happens to this day. We see infections now and then. This, by the way, is the range of the flying fox, that bat I just showed you. So within this area, all these countries have the flying fox, and therefore are possible harbors of Hendra, and the next virus we'll see Nipah virus. But there is a vaccine for horses. It does not make sense to vaccinate humans. There aren't enough cases. There are lots of horse cases. So you vaccinate horses and you will also prevent human infections. This is called a one world health approach. The idea is that the world is all linked. All the living things on the planet are linked. So if you treat one, you can treat many. One world health. All right, next was Nipah, outbreak in Malaysia in 1998. So initially, here's Malaysia. Here's the Perak district. They have a lot of outdoor pig farms there, which they use for food. And on one of them, there was a huge outbreak of disease in pigs, respiratory and neurological disease on pig farms. And the only way they knew to stop the infection was to kill 1 million pigs. And 105 humans died also. They were the people who worked with the pigs. How do you kill a million pigs? Do you know? Do you inject each one lethally? That would take you a long time, wouldn't it? They bury them.
They bury them alive.
They bury them alive. There's a big pit. You can see there's a video on YouTube, and that's so sad. They're just pushing the pigs in and burying them. Anyway, that stopped the outbreak. There were some cases in Singapore because they shipped pigs from Malaysia to Singapore, which is way down here, and that outbreak was controlled also. So what happens here is that, once again, fruit bats, flying foxes or fruit bats are infected with the virus. They excrete the virus in the urine. The pigs get infected what happens is often the farmers will plant mango trees in the piggeries. Piggery is the name for the pig farm. And the bats at night will eat the mango, and they will drop it on the floor, on the ground, and the mango is now contaminated with Nipah virus. And then the pigs eat the mango and they get sick. These infections still occur, no longer from pigs, but from consuming date palm sap. Date palm sap is sap taken from a palm tree, a date palm tree. People love to drink it in India and Bangladesh and many other countries. And a risk factor for getting Nipah infection turned out to be date palm sap. So what you do to make date palm sap is you tap the tree and you collect the sap over days into a container. It turned out at night the bats were going in the container and drinking the sap and contaminating it with virus. So they just covered all the traps and that stopped that transmission. But there's still infections and we're not sure what's going on. Now we know, this has been studied extensively, and we know why there was this outbreak of Nipah on the pig farm in Malaysia originally. So here's the host, the flying fox, natural reservoir flying fox. There was a lot of deforestation going on in that part of Malaysia. At the same time, there was an El Nino event, which caused drought. The forest fires that were being pushed for deforestation called haze. All of this reduced the flowering and fruiting trees that flying foxes need for food. So what do? They left their environment and found mango trees in the piggeries. And that's how they delivered the virus. So we did this by doing the deforestation to build farms. We caused these bats' habitat to change and led to an infection of pigs and humans. Now, so as I said, these infections continue to occur. Originally, the outbreak was the flying foxes forage on fruit trees on pig farms. Pigs are exposed to material dropped by bats. Pigs get infected, rapid pig-to-pig transmission, pig to human. So we think we've stopped that. But now we have date palm sap being consumed by people. If they're not covered at night, they will get sick. And in many countries, you bury your own dead, so you prepare the body, and so that's a risk factor for getting infected as well. Then there have been some cases of Nipah which don't have a pig farm as a risk factor. They don't have date palm sap as a risk factor. We're not sure how they got infected. Horses can be infected as well. Humans can be infected after handling the horses. So these are continuing. And in fact, there's a Nipah vaccine that is in testing that can be used in case there is a big outbreak. This one I really like. This is Argentine hemorrhagic fever caused by Junin virus. So after World War II, Argentina decided to clear the pampas, these lovely grassy areas, to grow corn, maize. So they used herbicides to kill off all the grasses and weeds, and then they could grow corn. But then in the shade of the corn, a new shade-tolerant grass grew that wasn't there before. That grass was eaten by a new mouse, the Calomus musculinus, which happened to carry Junin virus. So the mouse wasn't there before because it wasn't the right vegetation for it. But when we changed the vegetation, the mouse came in, and then as people went in to harvest the maize, they got Argentine hemorrhagic fever, first isolated in 1953. Again, the stuff we do is responsible for us getting infected. All right, Ebola virus. Since 1976, we have had multiple outbreaks of Ebola virus. This is a Filovirus, an envelope filamentous virus with a negative stranded RNA genome. First infections in 1976 in DRC. And these spheres are roughly proportional to the size of the outbreak. You can see there have been multiple outbreaks in DRC, Uganda, et cetera, of different sizes. And then the big one in 2015, over 25,000 cases, causing a lot of death, which was in West Africa, in a place where it had never been before. So these dots are where these outbreaks have been. Mostly Western, Central Africa, and one outbreak in South Africa. And there are 4 different kinds of Ebola viruses: Zaire, Sudan, Typhus, and Bundibugyo. So these are spillovers. These are zoonotic. Each outbreak is a new zoonotic spillover. So humans are infected. This is a classic zoonosis. The index case, the first case, has some kind of contact with an animal carcass. They're hunting meat in the forest, and it's contaminated with Ebola virus. And then these people get sick. They transmit it to their family members, and hospitals end up transmitting it, taking care of patients. But the chains of infections are short, and they eventually stop. You can see all the outbreaks are relatively small. The reason the West Africa one was so big is because for 3 months they thought it was malaria, and they just let it fester. And then it was very difficult to control. So here's some examples. 1996 in Gabon, 37 cases. A chimp found dead in the forest was eaten by people hunting for food. 18 people involved in the butchering got ill. 10 other cases occurred in family members. So that's a classic zoonosis. Another one, 60 cases in Gabon. The index case was a hunter who lived in a camp. A dead chimp was found in the forest at the time he was infected. Now, the 2015 outbreak, as I said, was huge. This is the epidemiological tracing. So it began in a small village in Guinea, here, Meliandou village. There were 9 deaths from December 2nd, 2013 to February 8th. First recorded case was a 2-year-old child who had fever, black stool, vomiting, and onset December 2nd, died 4 days later. He transmitted the virus to his sister who died, to his mother who died, to his grandmother who died, to a nurse who took care of him, to a village midwife who died, but before that she went to another village and infected a lot of people. So you see how this happens, these chains of infections through family members and healthcare. And this slowly goes unnoticed for a long time, and you have a very large outbreak. We don't know what this first case was doing. There's no... By the time the kid was sick, you couldn't really ask them anything, and so it's not clear what they had, what risk factor they had for getting infected. We'll never know. All right, so Ebola virus is a filovirus. It's the family of viruses, filoviruses. And the other member that infects humans regularly is Marburg virus. There were recent outbreaks of Marburg virus. Now, Marburg virus has been isolated. Infectious virus has been isolated from cave-dwelling fruit bats, Rosetus aegypticus. So it's clear that that's the reservoir for the virus. But we have never isolated infectious Ebola virus from any bat. So all we have is antibodies and some RNA in a couple of bats. So we do not consider that bats are definitive hosts of Ebola viruses. Yes?
Are there campaigns about warning people about eating bushmeat?
Yes. Also about how to prepare your dead for burial. There are certain practices that they're taught to do, and that really reduces infection. Yes. But now we have a vaccine, so if there's an outbreak, you can vaccinate people in the area, and that's really good. So maybe bats for Ebola, but humans, gorillas, and chimps are dead-end hosts. They get the virus and have a dead-end infection. I call it dead-end, even though there were 25,000 cases in Western Africa. Eventually, it fizzled out. It could not sustain human-to-human transmission, so that makes it a dead end. All right, so we think that bats are the reservoirs of these viruses, and periodically they spill over into chimps, gorillas, and by the way, the chimps and the gorillas die. So many gorillas have been lost to Ebola virus infections. It's the reason for the declining gorilla populations in Africa. And then humans are involved in the spillover. Sometimes humans get it from bats. People do hunt bats for food, but sometimes they get it from chimps. Gorillas are unlikely because you rarely see live gorillas, and if they have died, then the carcass is gone, and you're not going to see it. So there is limited human-to-human transmission at close quarters. So contact with infected blood, body fluids from someone who's sick or has just died. So you can imagine in a family taking care of a deceased family member, you could easily contaminate yourself. It can be spread by needles, syringes, but it's not spread by insects, it's not spread by water, it's not spread by food, it is not spread by respiratory droplets. It has to be close contact. And so, for example, you have a patient with Ebolavirus, hemorrhagic disease, needs to be intubated. So the physician is intubating them, and when you put the tube in, often the patient regurgitates fluid into your face, and that's how you can get infected. SARS-1, 2003. There is a very nice email service called ProMedMail that tracks infectious disease outbreaks. And this is one of the emails from ProMedMail back in February from this fellow who said, "This morning I received this email and then searched your archives and found nothing. Does anyone know about this? Quote, have you heard of an epidemic in Guangzhou? An acquaintance of mine from a chat room lives there and reports that the hospitals there have been closed and people are dying." So this is February. This outbreak began in November, but China decided not to tell the world. And by the time they did, it had spread extensively. So this was SARS, severe acute respiratory syndrome. It was an outbreak of atypical pneumonia in the south. November 2002 started with 305 cases and 5 deaths, short incubation period, chills, headache, malaise during the prodrome, dry cough, shortness of breath, and then 10 to 20% of people require mechanical ventilation. Now, fortunately, this virus was not transmitted effectively until the peak of disease, not transmitted during the incubation period. And so that's why eventually it petered out. And so a Chinese doctor who treated patients in China, in Guangzhou, traveled to Hong Kong on February 21st. He stayed at the Metropole Hotel. This is a very famous hotel now because of SARS-1. He died the next day in the hospital of SARS. But he had spread already the infection to 10 other people in the hotel who flew to other countries, Singapore, Vietnam, Canada, the US, before symptoms were evident. And eventually the infection spread to 8,000 people in 29 countries, 10% mortality rate. This is a tourist poster from Hong Kong. Hong Kong will take your breath away. If you get SARS, it will. So this is the epi curve of SARS. So 8,000 cases, most of them in China and Hong Kong, as you can see, 10% case fatality. And again, the first reporting was in February, it peaks around March, and the outbreak ends July 2003. So this is a coronavirus related to SARS-CoV-2, but still quite different. So where did it come from? Here's some of the data. Human sera collected before the outbreak have no antibodies to SARS-CoV. So this is a brand new virus. But the earliest cases in Guangdong were in handlers of animals for the exotic food market. So these open meat markets where you can see there's meat hanging there that you buy, or there are animals that you can pick and are slaughtered. And that was the source of the virus. So these early handlers had higher anti-SARS-CoV antibodies than control groups. What happens then subsequently for the next 10 years, they were looking for the ancestor, and they found in a cave in Junin, about, I think it's 1,000 kilometers away, all the elements to make SARS-CoV and different viruses that would have to combine to make SARS-CoV. They never found the direct progenitor, but they found all the viruses, when recombined, would make it. Now, during the outbreak in this province where the cave was, Junin, there were no cases. Why wouldn't there be cases if that's where it came from? So the idea is it emerged in bats in Junin and then infected an intermediary animal, the civet cat, which were then transported to the Guangdong markets, and then they infected people. So there were no bat to human transmissions. It went bat, civet cat to humans. So that's the horseshoe bat, which is host to many coronaviruses. You know why it's called a horseshoe bat? Because this looks like a horseshoe right here on its face. Those are its eyes, that's its mouth, okay? Nose. And this is a civet cat. MERS coronavirus emerged in 2012. A 60-year-old patient died of pneumonia and renal failure. They recovered the virus, sequenced it, found the receptor, found it was closely related to bat coronaviruses, but not SARS, so they gave it a new name, Middle East Respiratory Syndrome coronavirus. And the idea was that they eventually sorted out it is spread from dromedary camels, which are throughout the Middle East and Africa, as I've shown you, to humans. It doesn't transmit well among people, so it's usually a camel to human spillover, and a vaccine for camels is in testing because, again, there are not enough human cases to vaccinate people, but there are, every camel that is born on a camel farm gets infected but doesn't have serious disease. So if you vaccinate the camels, you may be able to protect humans. Most of the cases have been in the Arabian Peninsula. There have been some cases exported to Africa. South Korea was a famous one. This guy went to South Korea. He got sick. He was admitted to the hospital. I had no idea what he had. And in the meantime, the hospital worker spread his infection to a bunch of other patients. And then they figured out that he had MERS coronavirus. So most of the transmissions are camels to humans, very little human to human. And this is the number of cases, 2,468 and 851 deaths in 27 countries as of a couple of years ago. Now, I just want to show one slide or two of SARS-CoV-2 because we'll talk about this next time in more detail. The third pandemic of the 21st century, SARS-CoV-2, 2009 H1N1, HIV/AIDS. It begins in Wuhan, December 2019, with a cluster of unusual pneumonia cases. Very alert physicians said, you know, the early cases are around the Huanan seafood market in Wuhan. So here's the Huanan seafood market. It is across the river from the Wuhan Institute of Virology here. And this was the early epicenter of the outbreak. All these dots are cases early on in 2019. You can see they're clustering around the Huanan market. And there are two different lineages that have merged, so two different spillovers from animals to humans. And we'll look at that in more detail next time. In contrast, there's no epicenter around the Wuhan Institute. And people say, oh, the virus escaped the lab in Wuhan. It can't be, because there's no epicenter. There's no evidence that it was there. And it's ridiculous that people don't understand the science. Well, it's become a political issue now, you see, and people ignore the science. You know, the biggest evidence for that people say that it was made in a lab is that they were doing gain-of-function research, right? Gain-of-function, yeah, right, that really proves it. That's not evidence. Anyway, 704 million cases. We don't count cases anymore for the last year because there's very little reporting. So that's as a year ago, 7 million deaths globally. We'll talk more about this next time. So the coronaviruses all came from nature. There are a whole bunch of human common cold coronaviruses, SARS coronavirus, MERS, SARS-CoV-2. There's even a coronavirus of pigs, SADS coronavirus, severe acute diarrhea syndrome coronavirus that went from a bat to a pig. You know, all these others went from bats or rodents to perhaps other intermediate animals to humans. Bats, camels for MERS, and bats, we don't know what the intermediary animal is. We'll talk a little bit about that next time, to humans for SARS-CoV-2. These viruses are very dangerous for the most part, Ebola viruses and Lassa virus. So you have to work under the highest containment, biosafety level 4. There are four levels, BSL 1, 2, 3, and 4. BSL 1 is your high school. There's nothing. BSL 2, you just close the door, you wear gloves and eye protection. BSL-3, you wear a respirator suit, self-contained, and that's what you needed for SARS-CoV-2 for a long time. And BSL-4 is the highest. You see, you have to wear a spacesuit that's connected to air. And as you move around the room, you have to disconnect and reconnect to different air sources. That's me. I visited the one in Boston, and we made a documentary, which is the only one possible because it was not open yet. It's very interesting to get into these suits. Before you put it on, you have to make sure it has no air holes in it. You have to fill it up with air and look for leaks, because if there's a leak, you could die. Anyway, BSL-4, high mortality viruses, person-to-person transmission is possible, and no approved vaccine or antiviral. There's none in New York City. It's not permitted. We have a few BSL-3s, and there's one BSL-4 in Boston, which is this one. So how common are these host range jumps? I already told you they're very rare, although they're rare in causing an epidemic. Dead ends are probably happening all the time. The ones that make sustained transmissions are very rare. Can we predict them? No, we cannot predict them. We don't have the ability to do that, but we can know what viruses are out there in species that have contact with humans and whether they have human pandemic potential. What does that mean? They can replicate in human cells. That's the best you can do. They can bind human receptors, they can get in, and they can make infectious virus particles. So for H5N1, the virus doesn't fit that bill yet. So that's why we still have to be vigilant. And we can do screenings. So one of the proposed ways to deal with this would be to have screening centers in major transportation centers throughout the world, train local people to do the screening and screen for the viruses that people are bringing in and out, and in case you pick up something that you don't want. But for coronaviruses, we can make pan-coronavirus antivirals. We should have made them after SARS-1, but it went away, and so people said, coronaviruses are not a problem. So anytime anyone predicts stuff like that, don't believe them, because you can't predict what viruses are going to do. All right, so next time we're going to talk about HIV, AIDS, and COVID pandemics, how they emerged, and what they're doing right now.